Hi, good afternoon, everyone. So today we will continue with week four's tutorial session, and again we will go through week four's tutorial content. Week four's summarize week four's content, and then look at few questions. So one more thing is there that this week what we had was so this week what we had. Like the week's content had nucleic acid, like looked at nucleic acid content, different types of biomolecules. One was nucleic acid characteristics, etc. And there was topics about sessions about on carbohydrates and lipids. So today we will cover carbohydrates and nucleic acids. Next session we will cover proteins and lipids. Okay. And also so the week four assignment is due on, I think, 18th February. So we will look at few questions, not the direct assignment questions, and we will also cover some of the concepts asked in the questions, so that can help you guys to finish the assignment. The MCQs. I will start sharing my screen then. All right. So to begin with, we will look at nucleic acids. Okay. So so the very first thing is, what is the composition of nucleic acids? So in general, nucleic acids are like ribonucleic acids, deoxyribonucleic acids, based on what is the composition of this particular nucleic acid in question. And also the week's content, the lecture video did have how from Griffith's experiment of transforming principle, that DNA is actually the genetic information. It was carried out using Staphylococcus aureus strains, different kinds of strains, heat killed strains, like rough strain and smooth strains. So either heat killed rough strain or heat killed smooth strains. These cocktails were injected into mouse, and then it was seen if they are dead or alive. However, even like in case of smooth strains, like even if the strains were heat killed, still where all the proteins etc are getting ruptured, still they could see that the mice was getting killed, which eventually gave the idea that okay, nucleic acid must be the transforming principle, which is actually kind of passed on, uh, remains intact, and it actually carries the encodes for the pathogenic entities. Okay. So now looking at the nucleic acid composition, it's DNA or RNA, and majorly composed of three components. It has a sugar, ribose sugar. It can be either ribose or as in pentose sugar, having five carbon molecules, and it can either be ribose. I think so. Someone wants to join the call. Okay. So, pentose sugar, it can either be ribose or deoxyribose, based on what is the composition at the 2' end. Okay, 2' carbon. So, if the 2' carbon is attached to the OH, attached to OH group here, then it is ribose. And if this O is missing here, it is simply H here, then it is 2' deoxyribose sugar. So one is the pentose sugar and another is the phosphate group or phosphoric acid, and another is the base in question. So the bases can be um, the bases can be either purines or pyrimidines, and purines are adenine or guanine, and pyrimidines can be thymine, cytosine, uracil. Uracil is present in case of ribonucleic acids or RNA, and uh, like thymine in case of deoxyribonucleic acid or DNA. Okay. So the phosphoric acid provides the backbone to the polymer, whereas sugars, these pentose sugars, work as the anchoring point for nitrogenous bases. And the 9-membered nitrogenous bases here, these ones, give diversity in the sequence of the nucleic acids. Okay. Eventually, this combination gives the genetic codes and codons as well, which eventually codes for different kinds of amino acids to give rise to proteins. Now, if we look at the nucleic acid structure, nucleotide, if we call like what is a nucleoside, so usually nucleic acid has pentose sugar, phosphate group attached to it, and a base attached to the sugar. Okay. So nucleoside is basically only the base plus the sugar. Nucleoside monophosphate, like, in general, nucleotide is the base plus sugar plus the phosphate group attached, triphosphate group attached to it. Okay. And nucleoside, only the base and the sugar. The sugar can be either ribose or deoxyribose. Nucleoside monophosphate, the nucleoside is attached with one phosphate group, nucleoside diphosphate or nucleoside triphosphates, NTPs. Okay. So nucleotide has a free hydroxyl group OH at the 3' carbon. Here, this one is being talked about, and a phosphate group at 5' end of the sugar moiety. So this is the ribose sugar. Here the base is connected at the 5' end, the phosphate group is connected, and 3' end there is OH group. Okay. So the first nucleotide has a free phosphate at the 5' end here, this one connected to this 5' carbon. Okay. And the first has a free phosphate, and the 3' carboxyl group, 3' hydroxyl group at the 3' carbon position makes bond with the phosphate group at the 5' of the next nucleotide in question. Okay. The propagation of nucleotide along the length of the chain gives rise to a polynucleotide. As a result of each polynucleotide chain, a free 5' phosphate group on the first nucleotide, this one, and a free 3' hydroxyl group on the last nucleotide, this one, like once consecutive nucleotides are joined. Eventually, we have a free 5' phosphate group and on the first nucleotide and 3' hydroxyl group on the last nucleotide. It gives the polarity to the polynucleotide chain and thereby running in the direction from 5' to 3'. Okay. Now, definitely DNA is a double-stranded helix, and RNA is single-stranded, and both DNA strands or the helices are held together by hydrogen bonding between the bases attached to the sugar. Purines and pyrimidines, basically AT and GC. Adenine pairs with thymine with two hydrogen bonds. If we see here, thymine in case of DNA and uracil in case of RNA. Okay. So adenine, this nitrogen, this hydrogen attached to this particular N residue forms hydrogen bonding interactions with the O here, double bonded O at thymine, and this particular N here again forms hydrogen bonding interactions with the H attached here on thymine. Okay, so there are two hydrogen bonds. Um, and here in case of guanine and cytosine, you have three hydrogen bonds. This one here, and this one here. Okay. So adenine of one chain is always making two hydrogen bonds with the thymine of the next chain. Similarly, guanine of one chain is making three hydrogen bonds with the cytosine residue of the next chain. Okay. and we will look at the pitch and rise of the helix as well. So if we talk about, this is the DNA helix, this is the major groove. Okay, this is the major groove and this is the minor groove here. And if we talk about the axis rise, which is usually 3.4 angstrom, and helix diameter is 20 angstrom approximately, and one helical turn, you have 10.5 base pairs per turn, and the helix pitch, one complete helix, if we talk about, is approximately 35.7 angstroms. Okay. Now, why such base pairing, that purine, like adenine to thymine, guanine to cytosine? Because adenine or guanine is a purine base and has a 9-membered structure, whereas thymine or cytosine is pyrimidine, which is 6-membered. Okay. Now, if we look at the presence of both purines, which are bulky side chains, or pyrimidines, which have smaller side chains, 6-membered rings, right? These make it difficult to accommodate or too short to form the hydrogen bonding within the DNA strands. So the structure that was like X-ray data that was there, it gave a like helix diameter, right? So based on that, if we had two purines or two pyrimidines together, it didn't fit in with the experimental data. So eventually, if a purine and a pyrimidine were there, then it was a perfect match for hydrogen acceptor as well as donor sites, as well as the size, dimension wise also, they fit well within the with the X-ray experimental data. Okay. And anything else I'm missing? So if the pairing is ATGC, so that is what is also known as complementarity. Okay. So this is base pairing and it is a complementarity there, with adenine pairing with thymine residue on the complementary strand, and guanine on one strand pairs with cytosine on the complementary strand. Now, the purine and pyrimidine bases are hydrophobic and relatively insoluble in water at a near neutral pH. Okay. Now, when the pH either decreases, acidic, or increases or becomes alkaline, the bases become charged and their solubility in water increases. Okay, because uh, there are nitrogen and oxygen residues, right, on the bases. So, based on that, depending on their surrounding polarity, of the polarity of the surrounding medium, the charges, overall net charge on the basis, whether 9-membered ring of purine or 6-membered ring pyrimidine basis, the charges vary, and purines and pyrimidines are highly conjugated molecules. You saw, so here you have double bonded regions here, right? So eventually these electron clouds in the double bonded regions can actually delocalize, and eventually there's an electron cloud delocalized over this complete area. Okay. So purines and pyrimidines are highly conjugated molecules, and resonance, or that uh, the electron cloud getting delocalized all over that planar area, uh, this resonance leads to like giving most of the bonds a partial double bond character, right? And as a result of resonance, all the nucleotide bases absorb UV light, and nucleic acids are characterized by a strong absorption at wavelengths at nearly 260 nanometers. So in general, whenever we looked at plasmid purification the previous day, right? I mean, in the previous tutorial session. So there, in general, whenever we are purifying any kind of nucleic acid, whether it's RNA or DNA, usually it's to see the content of the like what is the amount or the integrity or purity of the nucleic acid of the nucleic acid sample. We always measure the UV at 260 nanometers range, where these bases having double bond character or partial double bond character, rather delocalized electron cloud all over the planar region. So there, due to this particular characteristic, it absorbs at 260 nanometers, and we can eventually check the purity or the content of, or the amount of the nucleic acid in our sample. Okay. So each base pair of DNA is twisted or displaced from the previous one by an angle of about 36 degrees. And if we talk about free pyrimidine and purine bases, these bases can exist in two or more tautomeric forms depending on what is the pH of the surrounding medium. So if we talk about uracil base, for example, it occurs in lactam, lactame and double lactame forms, and these tautomeric states are in equilibrium with each other. Although one type of, suppose we have ketoenol tautomerism with the double bonded O region there, right? So it either it exists as a ketone double bonded O, or it becomes alcohol OH, right? So these can interchange among themselves, although suppose in some cases keto form is more predominant than the other. So if we talk about the nitrogen atoms which are attached to the purine and pyrimidine rings are in the amino configuration and only rarely take the imino configuration. Likewise, the oxygen atoms are in the keto configuration and only rarely assume the enol configuration. The capacity to form tautomer is a frequent source of errors during DNA synthesis. Okay. So these are some of the tautomers. So this is the keto form. If we talk about thymine residue, this is the keto form, which is very commonly found. And sometimes this keto form can eventually give in rise to enol form. So here there is double bonded region between, I mean double bond between C and N in case of ene, like ENE double bond, and then here OH becomes, so it becomes enol, alcohol, and ene here. Okay, double bonded region here, so it is the enol form. Although this is the most common form, the keto form. Again, in case of adenine, if we talk about adenine, you have this commonly predominant form, which is the amino form. However, the rare form is again the imino form, where this single bonded NH2 here becomes double bonded. Hmm. And then you have cytosine as well. Again, it can show amino or imino forms, like changes between amino and imino form, again where amino form is the commonly found form, commonly existing form. And again, guanine can show ketoenol tautomerism, where again the keto form is the more common form, more commonly existing form, where again this region, this sixth carbon here, can actually either exist as C double bonded O form or C double bonded to N, ene, and attached with an alcohol group OH, here giving the enol form. Okay. So either amino to imino tautomerism or ketoenol tautomerism. Okay. Now these bases that we are talking about, purines and pyrimidines, tend to stack above each other roughly perpendicular to the direction of the helical axis. Okay, so this is also called as base stacking interactions. And hydrophobic interactions and van der Waals forces between the stacked planar bases, base stacking interactions provide stability to the entire DNA molecule. And these helical turns and planar base pairs cause the molecule to resemble a spiral staircase. Okay, so the DNA double helix that we are talking about resembles a spiral staircase, and mostly the stability is given by the hydrogen bonding interactions, the base stacking interactions, and then the overall van der Waals forces and hydrophobic interactions as well. Now, the two strands are held together by hydrogen bonds between bases of opposite strands or complementary strands. And because individual hydrogen bonds, if we talk about, this is not a covalent bond, right? These are all non-covalent interactions. So, these individual hydrogen bonds are weak and easily broken, and the DNA strands can become separated due to various activities or due to different kinds of environment, whether uh, there's a change in temperature, change in pH, etc., or upon exposure to some kind of radiation, then the DNA strands can actually, I mean DNA strand, like complementarity between the two strands can actually break. But the strengths of the hydrogen bonds are additive, and the large number of hydrogen bonds holding the two strands together make this DNA double helix a thermodynamically stable structure. Okay. On top of that, you do not have the OH group at the 2' end of the ribose sugar, right? I mean, sorry, pentose sugar. So giving it a having a deoxyribose sugar that also gives an additional stability to the DNA molecule as compared to the RNA molecule. Okay. Definitely the two strands are there, which gives it a stability, as well as the absence of the uh, O atom at the 2' end of the ribose sugar, um, pentose sugar, giving rise to deoxyribose sugar, adds into the stability of DNA molecule. Now, if we talk about the properties of nucleic acids, the effect of pH and temperature on nucleic acids, um, the nucleic acids are stable between a pH range of 5 to 9. And if we kind of go beyond this range, they undergo denaturation spontaneously. At low or acidic pH, the bases become protonated because of more concentration of H plus in the surrounding medium, and thus become positively charged, thereby repelling each other, right? And at high or alkaline pH, the bases lose protons and become negatively charged, again repelling each other because of similar charges. Now, when we talk about temperature treatment, when the temperature increases, the resulting increase in molecular motion eventually breaks the hydrogen bonds between the complementary bases and other forces that stabilize the double helical structure. Hence, the strands separate, driven apart by electrostatic repulsion of the negatively charged deoxyribose phosphate backbone of each strand. And near the denaturation temperature, a small increase in temperature causes a rapid, near simultaneous loss of multiple weak interactions holding the strands together. Okay. And definitely when nucleic acids denature, especially DNA, denature, RNA can be single stranded or double stranded as well. Okay. So density of nucleic acids increases upon denaturation. Effect of acids and alkali on nucleic acids. If we talk about DNA and RNA, both can undergo acid hydrolysis as well as alkaline hydrolysis. RNA readily undergoes alkaline hydrolysis, uh, but DNA is relatively more stable than RNA to alkaline hydrolysis due to the absence of 2' OH groups in the sugar of DNA. If we talk about effect of bisulfite and nitrous acid, so bisulfite and nitrous acid promote deamination or removal of an amino group from cytosine, resulting in the conversion of cytosine to uracil. In DNA, cytosine deamination will ultimately lead to conversion of a GC base pair to an AT base pair if left uncorrected. And if we talk about the effect of ionizing radiations and oxidizing agents, these actually non-specifically damage DNA by breaking the phosphodiester backbone and producing polynucleotide fragments with different 5' and 3' terminal products. They also directly attack and alter the chemical composition of bases in the nucleic acids. And many DNA molecules are circular as well. DNA in eukaryotes exist in the linear form, whereas we looked at all this in the previous tutorial as well, whereas many prokaryotic and viral DNA are circular molecules. And circular DNA molecules are also found in the mitochondria and chloroplasts. Okay. Now, if we talk about the chemical instability of RNA at alkaline pH, what actually happens is, suppose this is an RNA strand linked by phosphodiester linkage here. This here ester linkage, and this here is another ester linkage. So this is there, and then when OH minus, so at alkaline pH, there is an increase in the concentration of OH minus. Now what happens here is it has 2' OH group present here in the this ribose sugar has 2' OH group here. So this OH minus actually tends to attack this oxygen here and take away the H plus here, and eventually this attacks this phosphorus atom here, and this bond is again O eventually has a lone pair here, and finally what happens here is this overall nucleotide goes off and eventually a shortened RNA molecule. So this has continuous nucleotides added after one after the other after this particular area. Okay, this there are further phosphodiester linkages downside. And however, upon alkaline attack or like in an alkaline medium, this particular nucleotide goes off, okay, giving rise to 2', 3' cyclic monophosphate derivative, and a shortened RNA strand, and eventually you have a mixture of 2' and 3' monophosphate derivatives. Okay. So next, how to really isolate genomic DNA. Okay. Overall, if we look at genomic DNA, so we have to eventually take something out of the cells. So definitely the very first thing is to break the cells open, which is the lysis step. Then you have to treat the overall content with certain enzymes so that you precipitate out. Suppose in this case, we are talking about genomic DNA purification, right? So we don't need proteins or RNA molecules, etc. In our sample, right? We need pure genomic DNA. So for that, different kinds of treatments are there, and eventually, like some enzymatic treatments are there. You then again treat it with alcohol, chloroform, phenol, and finally there's phase separation and all. And then you have you precipitate the DNA with alcohol and solubilize in water. Now we will look eventually at each of the steps. Okay. So if we talk about lysis here, okay, maybe one by one we'll go. So, lytic step can be of many types. There can be mechanical lysis or bead beating stuff, and then there's sonication and stuff. So, there are different ways to lyse, break the cells open. So, one is simple mortar and pestle, like we find in Indian kitchens, and so which is actually you grind the complete cell mixture, often used for plant samples, frozen in liquid nitrogen since the specimens are comparatively bulky. So, what usually happens in case of plants, right? So, suppose you want to isolate the genomic DNA from leaves. Okay, so what if you take normally a fallen leaf or maybe pluck a leaf from a tree or a plant, then crushing that particular thing won't be really feasible. So what happens here is if you put it in liquid nitrogen exists at approximately minus 192 degrees Celsius. So it will actually, make the complete thing, freezing it at such low temperatures will eventually make the sample very brittle. So eventually you can, even you can actually, rather than having soft tissues and all, you can actually very nicely freeze it uh, at such low temperatures and eventually crush it because it becomes brittle. You crush it and then grind it in using an appropriate buffer, and eventually that helps in grinding the cells here in case of plants. I was talking about plants. And then after lysis, metabolites and samples can be extracted by further downstream processes, where again the solvents, the buffers matter. Okay. But here we are focusing on the lysis step. Then there can be bead beating as well. So there are glass or ceramic beads like spheres. Okay. So those are there to crack the cells open. Nothing like you have the cell mixture and you put it. Suppose you have a beaker or a tube um, where you have put in glass beads or ceramic beads, this like spherical balls, and then you have put in your cell mixture, and eventually this is some like you vortex it at a very high speed or something, or you rotate it, let it rotate at whatever speed is feasible for your sample. And then eventually this mechanical pressure, this mechanical forces from the glass beads eventually cracks the cells open. Okay. And this type of mechanical shear is gentle enough to keep the organelles intact. And the bead beating can be used with all cell types. Eventually, what you simply do is what I said, you add beads to the required amount of cell suspension or mixture and vortex. Okay, or you can eventually, if you need to, purify some kind of really, um, what to say, you do not want to expose your cells to like very harsh treatment, so you do not generally vortex. You simply put it on a rotator, which there is 360-degree rotation of the complete tube. So you will have the beads at certain speeds, where it will rotate 360 degree and eventually it will crush your cell mixture. The beads present inside will crush your cells and you will have your intracellular components coming out. Okay. There can be sonication. Sonicators induce rapid vibrations in a titanium probe immersed in the cell solution. Eventually, what happens is cavitation, where tiny bubbles are produced within the cell suspension, and bubbles explode, and this explosion sends local shock waves which disrupt the cell wall. So eventually, like a bomb explosion, if we talk about, um, so an explosion, bomb explosion disrupts things in the blast radius, right? And in that case, the medium is air, right? But in this case, in case of sonication, the medium is liquid here, and it is happening at a very small scale also. Okay. Then there are homogenizers, which again use shearing forces, which eventually lyse the cells, almost similar to how bead beating works, and it can be performed by squeezing the cells through a tube that is slightly smaller than the cell dimensions. Now imagine suppose you have to like you have a very narrow passage through which the cells are passed, and the passage is actually narrower than the diameter of one cell. Okay, so eventually if you pass the like put in the pressure and pass the cells through that particular narrow passage, then eventually the outer coverings of the cell will be burst open. Okay, and the intracellular components will come out. So this will eventually shear away the outer layer. Okay, homogenizing method works in this way. Now there's high temperature lysis as well. So high temperature and pressure can break chemical bonds within the cell walls and eventually can also denature proteins. So which is actually a very quick but dirty cell lysis method. So microwave or autoclave to generate the heat and pressure. Usually this is to be avoided because this can actually denature your sample. Suppose you want to purify proteins and you are exposing the cells to high temperature lysis, then there is a high chance that your proteins will be degraded or denatured. Enzymatic lysis can be there, like which eventually breaks open cells using enzymes that degrade cell walls and membranes. Depending on the organism, one can actually use cellulases, right? Cellulose walls are there in case of plants, chitinases, bacteriolytic enzymes like lysozyme, which actually interferes with peptidoglycan uh, cross-linking. There can be manganese or glycanase. So all these, wherever in case of enzymes, if we talk about ases, wherever you have ases, so those are like degrading or catalytic enzymes, basically breaking that particular component in question. So chitinases, or if we talk about simply proteinases, so which are protein degrading enzymes, or nucleases, nucleic acid degrading enzymes. So similarly, manganese, so it will degrade the mannan compounds there, mannan metabolites there. Okay, so enzymatic lysis is actually useful if you want to isolate the protoplast, which is a cell without the wall and without the cell wall. Again, there can be you can use different kinds of chemicals like organic solvents, alcohols, ether, or chloroform to disrupt the cell wall by permeabilizing the cell walls and membranes. It is actually handy to extract hydrophobic molecules like plant pigments. Even one can use EDTA. EDTA is ethylene diamine tetraacetic acid, which is a chelating agent. So it chelates it chelates divalent ions, right? EDTA it chelates divalent ions by coordinate bonds, and eventually, so in general, these divalent ions like suppose calcium Ca2 plus, magnesium Mg2 plus, etc., or all these are actually very important components to maintain the integrity of the suppose proteins, or there are different kinds of cellular structures. So these actually hold together different kinds of molecules, different kinds of biomolecules together, and eventually help in the integrity of the complete structure, okay, formed by these biomolecules. So if we are adding a chelating agent like EDTA, so the bridges formed by these divalent ions can actually disrupt, and eventually this, if here we are talking about the cell walls, so again the cell wall's integrity will be disrupted, and and eventually you have to uh, you can actually break the cells open, remove the cell wall, break the cell wall, disrupt the cell wall, and you can have your cells out of the like outer layer. So, in case of gram-negative bacteria, cell walls contain lipopolysaccharides that are stabilized by cations like magnesium and calcium. And eventually, you should ideally dialyze the EDTA later, because now you have just broken the cell wall open, right? However, your complete cell is still there. Now, if you want to suppose purify a protein, or suppose in case of ribosomes, you want to really purify ribosomes. So ribosomes are ribonucleoprotein machineries, right? It has RNA as well as ribosomal proteins. And eventually the ribosomes, whatever the functional unit is, those are actually held together by different kinds of interactions, out of which one is the interactions with different kinds of ions, not only divalent ions, there are monovalent ions as well. So all these ions are very crucial part to maintain the integrity of ribosomal structure. Now if you want to purify ribosomes, now you have only the cell wall disrupted, and you have the cells out. Now if you still have EDTA remaining, then it will actually disrupt the integrity of ribosomal structures or any other protein you wish to purify, which is actually stabilized by the presence of certain ions, not necessarily divalent ions, it can be other monovalent ions as well. So essentially you should dialyze, which is to remove the EDTA later, so that downstream processes are not affected. Depending on, again, all this depends on what you wish to purify. Then there can be freeze thaw cell lysis. So what happens is, thawing is basically, I mean, melting the thing. So what happens here is freezing creates ice crystals, and water expands upon freezing, that melts when the sample is thawed. So several freeze thaw cycles ultimately can help in rupturing the cells. This is comparatively gentle, however, this is a very time consuming step, and it doesn't generate any heat, but it's really time consuming, and it will take a long time to break the cells open if we simply use freeze thaw cycles like consecutively. Now we looked at phenol chloroform extraction, right here, this step, purification with chloroform phenol. This is basically to remove the proteins. Okay, so how exactly does it work? So, phenol is added to the aqueous soup containing the proteins and the DNA to be purified. Okay. Now, phenol and water are immiscible. Eventually, it does not mix, and like the phenol and water do not mix and do not form a homogeneous mixture. So, you can see very distinct phases. One is the aqueous phase containing water, and one is the organic phase containing the phenol. And phenol being denser out of these two liquids, it prefers to sit at the bottom, and the phases are then actually mixed by vortexing, so it leads to a thorough mixing. Eventually the phenol layer is actually forced into water layer, thereby forming emulsion of droplets, right? And so what happens here is proteins in the water phase which are present are denatured and partitions into phenol layer, while DNA stays in aqueous phase. Now, why is it denatured? In general, like when we move into the proteins part, so proteins have hydrophobic regions as well as like polar regions as well as non-polar regions or hydrophobic regions. So the in proteins, what happens is the polar region loves to interact with the polar environment, and the hydrophobic regions, which are water hating or water fearing regions, try to stay away from water, right? So they eventually kind of are uh, hidden away from water, whereas the polar residues or the polar regions are kind of outside, interacting with the aqueous environment. Now what happens in this case is proteins are there happily there with the hydrophobic or water fearing part hidden inside, and the polar regions only interacting outside. However, upon phenol droplets, okay, what happens here is it eventually gets a very kind of the phenol is comparatively hydrophobic, right? So eventually the hydrophobic part tries to interact with the organic layer or the phenol layer, and hence the whole integrity and the structure is disrupted in case of proteins. So what happens here is, due to these droplets, emulsion of droplets, emulsion of phenol droplets in the aqueous layer, this clear layer, what happens here is the proteins get denatured, whereas DNA very happily stays in the polar region, okay, or the aqueous phase. So this is what I was talking about. Um, this is DNA, and this is a polar environment or aqueous environment. So what happens is the polar residues of the protein, this blue region, they are happily interacting with the polar environment outside, and this green region is the hydrophobic region which stay away from the water. Now, whenever eventually the environment becomes less polar, so the less polar residues which were hidden inside, they are flipped outside, and eventually it is denatured. Okay, and flipped outside, and eventually what happens is it tries to stay in stay in the non-polar phase or less polar organic phase, which is the phenol layer, and DNA happily stays in the aqueous layer. So eventually the proteins are separated from the DNA molecules. So proteins flip inside out because the less polar residues, which are usually hidden inside, now want to interact with the less polar phenol. So the polar residues on the outside of the proteins made them soluble in water here in this case. And the phenol induced folding changes forced the phenol favoring residues to the outside. In this case, the green one comes outside, the hydrophobic residues or the phenol favoring residues. So the proteins are now more soluble in phenol than the aqueous phase. So some very polar residues may flip to the inside of the globular protein to be shielded from the unsuitable new solvent environment, right, which is less polar. So in short, the proteins are permanently denatured by the new solvent environment provided by phenol. So phenol chloroform. So mixture of buffer saturated phenol and chloroform, usually close to 1:1 mixture is used for DNA purification. Sometimes other ratios can also be used for RNA purification, like isoamyl alcohol is sometimes added as an anti-foaming agent, but it is optional, like phenyl chloroform isoamyl alcohol mixture is there. So the density of the buffer saturated phenol is actually a little higher than water. So if aqueous phase contains enough salts or any other solutes to increase its density, then you can actually see a phase inversion. Like earlier we saw the phenol prefers to sit at the bottom because it is more dense. However, what happens in these cases when aqueous phase contains a lot of salt or any other solutes, the aqueous phase comes under the phenol phase. Okay. Now, if that is the case, then eventually in the previous scenario, what we saw here is, suppose DNA is in the aqueous phase, proteins are in this lower, denser phenol phase. So you can easily separate this layer out. But if inversion is happening, the aqueous layer where you actually you want to purify your DNA molecule, which is happily sitting in the aqueous phase, now it comes under the phenol or organic phase, then how will you really take the sample out, because it will eventually contaminate your uh, DNA sample preparation. Okay. So to avoid this phase inversion, chloroform is added, which is significantly denser than water, and it is added to the organic phase. It increases the overall density of that phase. It prevents phase inversion. Chloroform helps reduce the interphase, the fuzzy border between the two phases, populated by molecules that can't really decide where they want to go. So fuzzy border can be due to partially denatured proteins or DNA depending on the pH, or partially denatured DNA binding proteins that are still clinging onto DNA. Now pipetting off some of this material while removing the aqueous phase it doesn't really help, because it decreases the purity of the sample, and if we pipette out less volume and leave a major volume on the top of the organic layer, then eventually we'll have a low weight. So you have to really be careful and kind of balance between these two steps while doing phenol chloroform extraction. Okay. in order to purify DNA or RNA. Like for RNA also we use phenol chloroform extraction. The principle stays the same. Now sometimes oxidation of the phenol produces pink or brown oxidation products, and these can actually be harmful for the DNA or RNA products. It can actually nick the DNA and degrade RNA products. Okay. So most commercial phenol solutions contain an antioxidant to inhibit this oxidation, and phenol buffered at an acidic pH seems to be resistant to oxidation. It's a good move to move a portion of the buffer saturated phenol from the brown bottle to a clear bottle or tube to inspect it before extraction to see if there is any color change or not. Okay. Because if oxidation has happened, it can eventually hamper your DNA or RNA samples. Now you have your purified DNA. Now you want to really check that if that is the exact sequence, what is the exact sequence of DNA purified, or if there is any other contamination from any other uh, DNA samples or RNA samples or not. So eventually you have to sequence it. Nothing, you get the sequence of the DNA molecule purified. Okay. So one is Sanger method. So this method was originally developed by Frederick Sanger in 1977. What happens here is a single-stranded DNA is used as a template to synthesize a complementary copy with the help of polymerase and in the presence of nucleotides. Right? We looked at the complementarity like A, T, G, C like that. So the if one chain has A, T, G, C, then the other chain will eventually have A, T. If it is T, then it becomes T, A, C, G. Okay. So in this method, what happens here is template is used to synthesize a complementary copy with the help of polymerase and in the presence of nucleotides. So the polymerization reaction contains a primer and nucleotides, three normal nucleotides and DDNTPs. Okay. So in general, you have deoxy NTPs. Okay. However, it is both. So you remember in the pentose sugar, the 3' OH was used for phosphoester linkage, right? However, in this case, usually 3 normal nucleotides with 5' phosphate and 3' OH group are used, but one of the tubes, like there are 4 nucleotides, right? 4 different types of nucleotides. So one of the tubes has both the oxygen missing, like 2' OH is not there, uh, as well as 3' OH is missing. So eventually, if the O is missing there, then you won't be the 3' OH group is missing, then it won't be able to form the phosphoester linkage. Okay. So that particular nucleotide is called DDNTP or 2' 3' dideoxynucleotide triphosphate. Okay. Now, when DNA polymerase utilizes DDNTPs as nucleotide, it gets incorporated into the growing chain, but the chain elongation stops at DDNTPs due to the absence of 3' hydroxyl group. So in the typical sequencing reactions, 4 different DDNTPs, because there are 4 different bases A, T, G, C. So 4 different DDNTPs are taken into 4 separate reactions and analyzed on high resolution polyacrylamide gels. The ratio of NTPs to DDNTPs is adjusted so that the chain termination occurs at each position of the base in the template. So one is the A reaction, one is the T reaction, one is G reaction, one is C reaction. Now what happens here is, suppose the reaction stops at this particular area. So it encounters DDATP, suppose. Okay, so it will stop here. So then if this is the case, then this becomes A. If again you have, so eventually how you do is, so this is uh, a gel where the matrix is porous and you load your samples here. Okay, and eventually it moves according to the sizes. Now the greater or the bigger the size, the bulkier the groups, it will take longer to really move through the porous matrix. So eventually the bands that you see here, which are higher up on this particular matrix, then these are the heavier or bulkier regions. and the ones that are the fastest moving, that you see at the lower portions, are actually smaller in size. So you have to, if we have to sequence DNA, then we have to start from the smallest fragment, smallest region. So eventually you see this is the smallest fragment out of the different amplification that we see here. Smallest fragment here is this one. So you start from down to bottom to you, like slowly you start from the bottom and then you slowly move up eventually one by one. So you go from the bottom. This is the number one where it is found, it is stopped at the A reaction here. Okay, so the first nucleotide or the first base here is A. Eventually you move to the second uh, smaller one is this one, which is at this T region. So A is there, then there's T. Again, you see the next smaller one is again in this T region. So again, another T gets added. So eventually you move in this way, here, then here, then here, then here. This way, eventually you sequence each individual nucleotide on the DNA strand and you get the complete sequence. So this is called Sanger sequencing. Okay. Another sequencing method is there, which is the Maxam-Gilbert method. This was actually discovered by Alan Maxam and Walter Gilbert, which is based on chemical modification and subsequent cleavage of the nucleotides. So in this method, what happens? A 3' or a 5' radio-labeled DNA is treated with base-specific chemicals, which randomly cleaves the DNA at their specific target nucleotide. And these fragments are then analyzed on a high-resolution polyacrylamide gel and an autoradiogram is developed. The fragment with terminal radio label appears as band in the gel. So here it's almost similar. However, the reaction tubes are a little different. One is the G reaction, G plus A reaction, T plus C reaction, and C reaction. So what happens here is again you have this particular porous matrix here, which is the gel in question. You have your 4 different lanes pertaining to different sets of reactions, and you load your samples. It moves from top to bottom. The bottommost part are the smaller fragments and fastest moving molecules, and the bigger and bulkier regions stay on top. Okay. Now how will you really analyze this? So the fragment in G lane is read as G, whereas a fragment present in G plus A region, this particular lane, but absent in G. If we talk about this particular fragment, it is not present in G lane, but it is present in G plus A lane. So then it will be read as G. Okay. So this particular fragment, then it comes to G again. Here also we have to move from the bottommost part and then move up. Okay. So this is G. Now here, uh, like this particular lane has both these area, however one strand is missing here, so it becomes GA. Then the next smaller fragment is this one. It is present in T plus C, and so it becomes T here. However, here it is present in both the regions, so it becomes C here. Okay. So eventually this is how, similarly fragment in C is read as C, whereas fragment present in T plus C but absent in C is read as T. Okay. To get the DNA sequence, the band with the lowest molecular weight is read. This one here, how we were looking at the smallest band, then the next smallest, the next smallest. The band with the lowest molecular weight is read, followed by next band in the 4 lane. Okay, 4 lanes. So the G lane, the band is of the lowest molecular weight, followed by band in the A lane, etc. Okay, eventually you get the complete sequence. All right. Now we move to uh, carbohydrates, and again those are called saccharides. It can be monosaccharides. The carbon atoms are chiral center. This is how the general chemical formula looks like, and the cyclization happens. Then this is how it looks like. So the whole lecture video, so this is taken from the lecture video only. We are kind of summarizing the things here. So the carbon atoms are chiral center to give stereoisomeric sugar found in nature. In addition, the reaction between the first carbonyl group, this one here, and the fifth carbon gives cyclic form of sugar. This and this gives the cyclic form of sugar here. depending on where the OH is, the Wejen-Dash formula or Fisher projections, based on the conformation of the or configuration of the molecule, it can either be alpha D form or beta forms, either alpha forms or beta forms, okay, of the glucose molecule in question. And when we talk about monosaccharides, are only one molecule is there, whereas disaccharides, you have two such entities joined together by condensation reaction, like this particular, like the H from one molecule and OH from the other molecule can actually react, condensation happens, H2O is released, and eventually a glycosidic, this particular bond is called the glycosidic bond. Okay. The individual monosaccharides utilize their terminal hydroxyl group to form a glycosidic linkage to form disaccharides, and with the loss of water. With the addition of water, it can again be hydrolyzed to form individual monosaccharides. Different monomers can participate to give different disaccharide sugar. Like here, maltose is there, so it is formed by the condensation of two glucose molecules. Okay. And glycosidic bond or linkage can again be of two types. O-glycosidic linkage, when the hydroxyl group on anomeric carbon, the first carbon of a sugar reacts with an alcohol or another sugar, and eventually they can exist in disaccharides, oligosaccharides, and polysaccharides, the O-glycosidic bonds. Okay, here in this case. Whereas N-glycosidic linkage, the hydroxyl group on anomeric carbon of sugar reacts with an amine. They are present in nucleotides, RNA, and DNA. Okay. So we saw in nucleic acid structure, we saw that the pentose sugar is attached to the base. So in that case, it is N-glycosidic bond. Whereas in case of monosaccharides or two monosaccharides or disaccharides, polysaccharides, then you have O-glycosidic linkages. Okay. Polysaccharides, condensation or removal of water of different monosaccharides or disaccharides give rise to polysaccharides. So polysaccharides are the storage form of sugar and gets hydrolyzed to give monosaccharides, which eventually participate in metabolic reactions to produce reducing equivalents to provide energy via electron transport system. Now, as the anomeric carbon participates in the formation of glycosidic bonds, it has lost its ability to form linear form of sugar. Hence, it becomes a non-reducing sugar end, like non-reducing end. Whereas in case of disaccharides or polysaccharides, the terminal sugar has a free anomeric carbon, and hence it is called the reducing end. Okay. Now, when we talk about cellular metabolism, the role of ATP as an intermediate between catabolism and anabolism. So, catabolism is catalytic pathways which eventually breaks down molecules to produce energy. Catabolism is energy producing reactions, whereas anabolism is biosynthetic pathways which actually utilize ATP or energy to give rise to complex biomolecules. Okay. Or synthetic pathways, basically to synthesize some kind of biomolecule, or in general synthesize components in the cell. Okay. Now catabolic reactions transfer energy from complex molecules to ATP. Heat is released, eventually giving rise to simple molecules such as glucose, amino acids, glycerol, and fatty acids. Eventually, these are used in anabolic reactions which transfer energy from ATP to build complex molecules. Heat is released, and complex molecules like starch, proteins, and lipids are formed. Okay, so if we talk about the overall metabolic reactions that go on in our body, this is how it happens. Okay, so one of the metabolic pathway is glycolysis. Because we are talking about sugars here, talking about carbohydrates here, the we have to talk about glucose, which eventually is used to give produce ATP in our bodies. So if we talk about the glycolytic step, complete step, it is divided into two steps. One is the preparatory phase, which requires energy, and another is the payoff phase, which releases energy. Okay, so here um, if we talk about the preparatory phase, you have this 6 carbon molecule glucose, okay, which uses ATP to phosphorylate uh, one of the residues, one of the residues there, giving rise to glucose 6 phosphate here. The uh, sixth carbon here becomes phosphorylated, giving rise to glucose 6 phosphate, and utilizes one ATP molecule. And this step is catalyzed by hexokinase enzyme. Okay. Next, glucose 6 phosphate is isomerized to fructose 6 phosphate by the enzyme phosphohexose isomerase. Okay, this is how it looks like. This is fructose 6 phosphate here. Again, fructose 6 phosphate is further phosphorylated using another ATP molecule to give rise to fructose 1,6 bisphosphate. So the sixth carbon was phosphorylated, and here in case of fructose 1,6 bisphosphate, the first carbon is also phosphorylated, and the enzyme used is phosphofructokinase. Okay, so any the enzymes which if you see read about kinase, this word kinase, this enzyme is always a phosphorylating enzyme. Okay, so here one phosphorylation is happening, it is hexokinase. Hexo because it is actually phosphorylating a 6 carbon molecule. Then you have phosphofructokinase. It is basically again phosphorylation is going on a phosphate bound fructose molecule. So that's why phosphofructokinase. Okay. And then fructose 1,6 bisphosphate gets cleaved into. So again, till this process, till this step, you have 6 carbon moieties altogether, okay, in one molecule. But eventually, this fructose 1,6 phosphate, bisphosphate, gets cleaved into 2,3 carbon moieties. One is glyceraldehyde 3 phosphate and DHAP, or dihydroxyacetone phosphate. This conversion is catalyzed by triose phosphate isomerase. Okay, so you have in this step finally 3 carbon molecules. Okay, like 3 carbon molecules which are different, but however you eventually have 3 carbon moieties. Now the 3 carbon moieties eventually enters the payoff phase. Okay, so here what you have is, suppose glyceraldehyde 3 phosphate, 3 carbon moiety enters the payoff phase, where oxidative conversion of glyceraldehyde 3 phosphate to pyruvate uh, like takes place, and this is also coupled with the formation of ATP and NADH. So glyceraldehyde 3 phosphate eventually utilizes NAD plus and inorganic phosphate to give rise to NADH plus H plus. Okay, why 2? Because there are 3 carbon moieties, and this utilize this gives this is oxidation happening here, even and as well as phosphorylation happening here. So you have 1,3 bisphosphoglycerate molecule. Again, all these are 3 carbon moieties. Okay, so 1,3 bisphosphoglycerate is there. This is the molecule, and this 1,3 bisphosphoglycerate is dephosphorylated to give rise to 3-phosphoglycerate. It has 2 phosphate groups here, right? 1,3 bisphosphoglycerate. However, upon dephosphorylation, it gives off 1 phosphate, and this phosphate is actually taken up by ADP to give rise to ATP, and 3-phosphoglycerate is formed here. Okay. Now, this 3-phosphoglycerate changes to 2-phosphoglycerate. It is kind of an isomerization here. Okay, and 2-phosphoglycerate changes to phosphoenol pyruvate by releasing H2O, and phosphoenol pyruvate finally gets dephosphorylated and giving rise to ATP and pyruvic acid or pyruvate. Okay, all these are 3 carbon moieties. So the number 2 in the brackets shows 2 molecules each of all these 3 carbon moieties. Okay. Uh, So this is the glycolytic pathway. Now, if we talk about the balance sheet for glycolysis, you have 1 glucose molecule going in, 4 ADP and 4 inorganic phosphate molecules going in, and you have two ATP molecules going in the preparatory phase and two NAD plus molecules going in the payoff phase. Output is two pyruvate molecules, two overall ATP that we are getting out, and two NADH molecules that we are getting out eventually, the output of this glycolytic pathway. Next, if we talk about regulation of glycolysis, so suppose there are different kinds of proteins which are called transporters and specific to different kinds of molecules or like other proteins as well. Okay, or ions. So suppose there's insulin hormone in our body. So this particular hormone can bind to its receptor here in the cell membrane. Okay, this one here, this white colored one is binding to the insulin receptor here, this particular complex here, and the receptor eventually gets activated, and this activated receptor promotes the recruitment of glucose transporters, which are again proteins. So these are glucose transporters, nicely packed and ready to do its function. So these are glucose transporters which are recruited from the intracellular pool onto the cell membrane, and glucose transporters increase insulin mediated uptake of glucose into the cells. Okay, these are glucose molecules, these black ones, and these white circular moieties are not really circular, but we can say that half circles. These are semicircular moieties which eventually help in taking up the glucose molecules. And when insulin level decreases, glucose transporters can actually move from the cell membrane to the intracellular storage pool where they can be recycled. Okay, and vesicles fuse to form endosome here, and it is stored inside. And again, when there is a signal from insulin receptors upon insulin binding, then again these glucose transporters or glucose receptors are uh, kind of recruited onto the cell membrane. Okay, this is one way to regulate glycolytic pathway. And another glycolysis, like another regulation happens at this level, at the level of pyruvate kinase. Okay, so what in general pyruvate kinase does is it takes a phosphate from phosphoenol pyruvate to give pyruvate. Okay, the last step before formation of pyruvic acid in glycolytic pathway. Now, in general, pyruvate kinase, this enzyme has two forms. One is the phosphorylated form, which is the less active form, and the non-phosphorylated or the native form, which is more active. Now, when there is low glucose, okay, what happens is dephosphorylated pyruvate kinase, like the pyruvate kinase is dephosphorylated from the phosphorylated one, then eventually it becomes more active. So it activates the conversion of PEP to pyruvate and produces energy, right? However, when blood glucose is high, there is sufficient energy, and glucose molecules catalyzes the phosphorylation of pyruvate kinase. Okay, so this phosphorylated pyruvate kinase is less active and slows down the PEP conversion of PEP to pyruvate. Okay, this is the overall regulation, that is like kind of summarizing these points here, what I talked about. This is the picturization or schematic representation. Another, so again, the levels of glucose 6 phosphate, substrate level control is there. Then AMP, levels of AMP, citrate, fructose 2,6 bisphosphate, etc. ATP levels, definitely. All these are also kind of regulators of glycolytic pathway. Now, the pyruvate that we got at the end of the glycolytic cycle actually enters the Krebs cycle or the citric acid cycle, or TCA or tricarboxylic acid cycle. Okay, so what happens here in this case? Pyruvate oxidation and citric acid cycle takes place in the mitochondrial matrix. So the pyruvate, upon reacting with the coenzyme A, it gets oxidized. Pyruvate is oxidized to acetate with the formation of NADH. Just I guess this will be more clear. So pyruvate is oxidized to acetate with the formation of NADH plus H plus, and CO2 is released. Acetate is activated by combination with coenzyme A, yielding acetyl coenzyme A. So pyruvate was a 3-carbon entity, and acetyl coenzyme A is a 2-carbon entity. Now, this acetyl coenzyme A, the 2-carbon acetyl group and the 4-carbon oxaloacetate from uh, like the previous substrate upon at the end of this particular cycle, which is a 4-carbon moiety, and acetyl coenzyme A, which is a 2-carbon moiety, combine to form citric acid, which is a 6-carbon molecule. Okay, and citric acid is rearranged or isomerized to form isocitric acid. Then NAD plus is used to give rise to NADH plus H plus, and one CO2 is released to give rise to 5-carbon moiety, alpha ketoglutaric acid or alpha ketoglutarate. Again, another carbon moiety is released and NAD plus is reduced, giving rise to 4-carbon moiety succinyl coenzyme A. Alpha ketoglutarate is oxidized to succinyl coenzyme A with the formation of NADH plus H plus and CO2. So NAD plus is getting reduced, but this is getting oxidized. Okay. So we will be saying that alpha ketoglutarate is getting oxidized to succinyl coenzyme A. However, and NAD plus is getting reduced to NADH. So this step is almost identical to pyruvate oxidation. Now, succinyl coenzyme A releases the coenzyme A here. This is getting released and becoming succinic acid, which is again a 4-carbon moiety only, but this is coupled to energy production. The energy thus released converts GDP to GTP, which in turn converts ADP to ATP. Okay. Now this 4-carbon moiety succinic acid is oxidized to fumarate, and FAD is reduced to FADH2, and fumaric acid is formed. Again a 4-carbon moiety, and fumaric acid and water react to form malate, and then again another malate is oxidized to oxaloacetate, thereby reducing NAD plus to NADH, and oxaloacetate, this 4-carbon moiety, can again react with the acetyl coenzyme A to re-enter this citric acid cycle. Okay, so this is the TCA cycle in question, which takes place in the mitochondrial matrix. Now again, this cycle also has regulators in place, right? It also needs to be regulated at certain time intervals. So, okay, the balance sheet occurs, TCA cycle occurs in the mitochondrial matrix. There will be 2 cycles per glucose molecule because one pyruvic acid is 3-carbon moiety and one glycolytic cycle from 6-carbon membered, like 6-carbon glucose, gives rise to 2 3-carbon pyruvic acid molecules. 3 NADH produced per acetyl coenzyme A, 1 FADH2 produced per acetyl coenzyme A, 2 CO2 is produced per acetyl coenzyme A via decarboxylation, and 2 acetyl coenzyme A transforms into CO2. So this is how the total per glucose molecule looks like in case of citric acid cycle, the inputs and the outputs. Okay. Now regulation of Krebs cycle or citric acid cycle. There are 4 limiting steps in Krebs cycle and the points where it can be regulated. One is the conversion of pyruvate into acetyl coenzyme A. This region is the first step which allow the entry of sugar moiety into the Krebs cycle. Pyruvate dehydrogenase complex is allosterically inhibited by high ratio of ATP or ADP, NADH or NAD+, and acetyl coenzyme A versus coenzyme E. Next, limiting step is the first reaction of Krebs cycle, catalyzed by citrate synthase. It is inhibited by high levels of NADH, ATP, and succinyl coenzyme A. Okay. The third is isocitrate dehydrogenase, which is again inhibited by high levels of ATP. This one here, ATP, NADH, whereas Ca2 plus and ADP actually stimulate this particular step. Alpha ketoglutarate dehydrogenase is inhibited by succinyl coenzyme A, this one here, okay, and high levels of NADH, whereas Ca2 plus actually stimulates this conversion of alpha ketoglutarate to succinyl coenzyme A. Okay, so the levels of all these different moieties kind of inhibits or promotes the forward conversion or remains at one particular substrate. Okay. So Krebs cycle or TCA cycle is called the master regulator of metabolism. Why? It is centrally connected to the metabolic intermediates of carbohydrates, proteins, and lipid metabolism. Okay. So there are actually, if you see in the TCA cycle, you have several branching points at each step where each of the intermediate can is actually a participant of some other metabolic pathway. Okay. Like if we talk about lipid metabolism, it is connected to the Krebs cycle through the common intermediates like citrate is there. Okay, this one citrate is there or acetyl coenzyme is there. Okay, and when we talk about protein metabolism, it also shares intermediates like alpha ketoglutarate, this one here, which is eventually involved in glutamate pathway. As a result, in, alpha ketoglutarate oxaloacetate is there as well here. Okay. As a result, Krebs cycle can allosterically or via product inhibition regulate other metabolic pathways. In addition, it can redistribute the intermediates between metabolic pathways and hence help in conversion of sugar to either proteins, lipids, or vice versa. Okay. Now, all this we were talking about in case of, and again, you have these molecules, and finally, in the presence of oxygen, uh, you have the electron transport chain occurring, right? But in case of anaerobic respiration, suppose in case of lactic acid fermentation, what happens is you have glucose uh, uh, changed to converted to pyruvic acid via glycolytic pathway. However, this pyruvic acid is converted or fermented to lactic acid, and you eventually have just a net ATP gain of only 2 ATP molecule, net gain of 2 ATP molecules and 2 lactate molecules here, which are fermented. However, we see in case of aerobic respiration, you have a nice complicated pathway going on, metabolic pathway going on like TCA, which eventually the NADH and FADH2 given off by the TCA cycle enters the oxidative phosphorylation pathway or electron transport chain, which eventually gives rise to ATP molecules. Okay. So this is aerobic respiration versus anaerobic respiration. Aerobic respiration, this part, glycolysis, net gain to ATP, enters the TCA cycle, um, gives off CO2, again 2 ATP, and finally oxidative phosphorylation, 32 ATP here. So also the FADH2 and NADH given off in glycolytic pathway and Krebs cycle. So each of these molecules, when they enter the ETS pathway or oxidative phosphorylation in the mitochondria, in mitochondrial inner membrane, what happens here there is those are eventually NADH molecules give rise to 3 ATP molecules and FADH2 gives rise to 2 ATP molecules. Okay. And if we talk about anaerobic respiration here, glucose converted to glycolysis with the release of ATP gives rise to pyruvic acid. However, either it can lead to fermentation, alcoholic fermentation, where you have ethyl alcohol and carbon dioxide, or lactic acid fermentation giving rise to lactic acid as a product. Okay. Now, some example questions. So, which of the following is a cofactor of hexokinase? Now, when we talk about enzymatic functions, the cofactor is enzymes actually require certain, accessories for it to function uh, fully or perform its function properly. Okay, so in this case, it can either require some kind of organic compounds or ions, etc. In case of hexokinase, which leads to conversion of glucose to glucose 6 phosphate in the glycolytic pathway, it actually needs magnesium ions as cofactor for its functioning. Okay. Now, so some enzymes require cofactors to be active. So if this is the enzyme, there is an active site. So enzyme is protein only. Example is lysozyme. And cofactors are actually non-protein component of an enzyme. And cofactors can be organic molecules. In that case, it is called coenzymes. It can be inorganic ions like calcium or zinc or magnesium, etc. Cofactors can also be permanently attached, in which in this scenario they are called prosthetic groups here. Okay, so enzyme, which is the protein part, and the prosthetic group, which is permanently attached. Example is flavoprotein plus FAD. or it can be temporarily attached, which is a coenzyme which can detach after a reaction and may participate with other enzymes in other reactions. Okay. So in glycolysis, two ATP molecules are utilized by the enzymes dash and dash. So the answers will be option C here, hexokinase. First of all, two ATP molecules are utilized. So that means two ATP molecules are utilized. That means ATP is being used, or energy is being used to give off ADP, diphosphate. Okay. And phosphate is actually phosphate, one phosphate moiety is used. So that means the enzymes are phosphorylating enzymes. So uh, so these will be kinases. So the last option anyways goes off, and you have kinases here. Now if we talk about the kinase in question, uh, two ATP molecules, so we are talking about the glycolytic pathway here. So the option will be hexokinase and phosphofructokinase here, which are getting utilized. I mean, ATP is getting utilized in this case. Fructose 1,6-bisphosphate is broken down into glyceraldehyde 3-phosphate and DHAP by which enzyme? The correct option is option B, aldolase. Okay. Which of the following cell surface receptor is involved in the uptake of glucose? So the correct answer is option C, glucose transporters here, GLUTs. During the event of low blood glucose, the enzyme pyruvate kinase is, so when there is low blood glucose, the enzyme pyruvate kinase is actually less active and phosphorylated. So this is the same thing which we looked at earlier. Okay, the same slide from the previous one. So one molecule of GTP is equivalent to dash molecule of ATP. One molecule of GTP is actually equivalent to one molecule of ATP. Which of the following pairs positively modulate the conversion of isocitrate to alpha ketoglutarate? We looked at the regulators of Krebs cycle, right? Isocitrate is one of the isocitrate and alpha ketoglutarate, both are intermediates of Krebs cycle or TCA cycle. So the correct answer is option B, positively regulate the conversion, as in it leads to the formation of alpha ketoglutarate. So higher levels of ADP and calcium ions actually helps in the forward movement, as in forward conversion of isocitrate to alpha ketoglutarate. And so here, isocitrate upon reducing NAD plus gives rise to, like, gives off CO2 as well, leads to alpha ketoglutarate. Okay, this is the overall reaction that takes place in this sequence, right? So isocitrate dehydrogenase is activated by ADP citrate and Ca2 plus, but inhibited by, so this conversion is taking place, right? So if NADH, the converted product, is already higher, uh, like the levels are high, then this is actually a negative regulator. Okay. If NAD plus levels are high, then it will actually help in the conversion of isocitrate to alpha ketoglutarate. Okay. So that's all for today's session. We will meet again in the next session, and we'll stop presenting. Thank you for joining. We'll discuss lipids in the next session and proteins together. Okay, and look at few questions. I will end the meeting now. Thank you for joining.